Picture picture a landscape where the temperature regularly pushes past 50 degrees Celsius.
Right, that's like 122 degrees Fahrenheit.
Exactly. I mean, the air is so dry it literally cracks your skin. And the sand is radiating heat back up at you. It makes the horizon physically shimmer.
Yeah, an absolute furnace.
And in this environment, humans willingly run a foot race called the Marathon des Sables.
Oh, wow, yeah.
It's a 251-kilometer ultramarathon across the Sahara Desert. People are running for hundreds of kilometers in blistering heat. And, you know, if you take a step back and think about the pure biology of that, it just seems entirely impossible.
I mean, it really should be impossible because when you're exercising at that level of intensity, your cardiac output, like the sheer volume of blood your heart is pumping, it doesn't just increase by a little bit.
Right.
It increases 4 to 5-fold.
That's massive.
It is. And your skeletal muscles are suddenly demanding a 5 to 10-fold increase in oxygen and metabolic fuel. And the byproduct of all that metabolic activity is heat.
Right, because you're burning fuel.
Exactly. You're generating massive, constant heat inside an environment that is, well, already trying to bake you.
Which really brings us to the central mystery of our biology. Because if a machine revved its engine that hard, the hydraulic lines would just burst.
Oh, instantly.
Like, if you didn't have invisible systems kicking in instantly to compensate for that running, that massive spike in cardiac output would cause your blood pressure to rise so high that your blood vessels would literally just tear apart.
They would. Or, you know, your circulating glucose levels would just crash, which would starve your neurons of energy and put you in a coma.
Jeez.
Or the heat from your leg muscles would trigger hyperthermia severe enough to actually denature your cell proteins. You would effectively cook yourself from the inside out.
So the fact that a human can run across the Sahara, or like conversely, survive at high latitudes where seasonal temperatures drop by 70 degrees Celsius.
Like the freezing cold.
Yeah.
Yeah.
It points to just an absolute architectural masterpiece inside the nervous system.
It really does.
Yeah. And exploring that masterpiece is our mission today. So welcome to part 1 of a special, highly detailed 2-part deep dive into the neurobiology of survival.
Yes.
We're going to thoroughly explore the foundational principles of neuroscience that dictate how you stay alive. Today, we're looking at the foundational architecture, like how your body maintains stability, the central coordinating hub buried deep in your brain called the hypothalamus, the exact neural highways your brain uses to send autonomic commands down to your organs, and finally, how your organs talk back.
Right. And if you consider yourself a dedicated student of the mind, I mean, this is the machinery you need to understand. We're going to map out the invisible automated systems that regulate your body temperature, your blood pressure, your digestion, your energy levels.
All the background stuff.
Exactly.
Yeah.
These are the systems operating right now inside you at this very second without you ever having to consciously think about them.
So to really grasp how the brain controls the body, I feel like we have to figure out what it's actually trying to achieve. Like, what is the ultimate goal of all this invisible regulation?
Yeah, that's the core question.
And to define that, we need to look at a concept introduced by the French physiologist Claude Bernard back in the mid-19th century.
Yeah, Bernard was a pioneer because he completely shifted the focus from the outside world to the inside. He drew attention to this really profound idea of the body's internal environment, which he called le milieu intérieur.
The internal environment.
Right. And its core proposition was that maintaining the stability of this internal environment is the fundamental condition that allows for a free and independent life.
A condition for a free life. I mean, that phrasing carries so much weight because if you look at, say, a single-celled organism in a puddle, it has zero freedom.
Right. If the puddle dries up, it dies.
Exactly. Or if a cold-blooded lizard wants to digest its food, it has to physically go find a hot rock to lie on. Its internal state is just a total slave to the external weather.
That's the perfect contrast. By investing massive amounts of physiological energy into stabilizing our internal temperature and our blood chemistry, mammals basically bought their freedom from the environment.
Wow, yeah.
And building on Bernard's philosophical foundation, you know, an American physiologist named Walter B. Cannon came along in the 1930s, and he gave us the technical term we use today, which is homeostasis.
Homeostasis.
Yeah. Cannon used this word to describe the highly specific, active physiological mechanisms that maintain the constancy of your bodily fluids, your core temperature, your blood pressure, your oxygen levels, all of it.
Well, for a long time, the dominant way people imagined homeostasis working was this set point model, and Honestly, this is totally how I always pictured it, just borrowing heavily from human engineering.
Yes, very intuitive.
Like, think of a commercial airliner on autopilot, or even just a thermostat in your living room. If I set my living room thermostat to 72 degrees, there is a literal physical sensor on the wall taking a reading of the air.
Right.
And if the room drops to 70 degrees, the sensor detects that 2-degree error, fires a signal to the furnace, and blasts heat until the sensor reads 72 again. It's binary, it's clean.
Very clean.
And we have this intuitive assumption that somewhere in the human brain, there is a singular master sensor tuned to exactly 98.6 degrees Fahrenheit.
Yeah, it's a very comforting analogy because we like things to be visible and neatly categorized. We want a clear cause and a clear effect. But modern physiology has completely abandoned the set point model.
Wait, really? Completely abandoned it?
Completely. Decades of intensive research, looking at the molecular, cellular, and systems levels have completely failed to uncover any singular molecular basis for encoding a specific master number.
Are you saying the human brain does not have a singular 98.6 degree sensor?
It doesn't exist. There is no biological thermostat in your brain tuned to an ideal temperature, nor is there a master sensor for your ideal body weight or your ideal blood pressure.
But wait, if there's no central sensor comparing my current temperature to a programmed master number, how is my core temperature so incredibly stable? Like, if there's no error detector, what is driving the correction when I walk outside into the snow?
Okay, so to understand that, we have to replace the set point with something called the settling point model.
The settling point.
Right. To grasp this, imagine a mountain lake.
Yeah.
A river flows into this lake, and another river flows out of it. Let's say it rains heavily for a week.
Right.
The inflow of water increases drastically, right?
Okay, yeah.
And the lake gets deeper. But as the lake gets deeper, the hydrostatic pressure of all that accumulated water increases.
Because it's heavier.
Exactly. And that increased pressure physically pushes more water out of the outflow river at the bottom. Eventually, without any intelligent intervention at all, the increased outflow naturally matches the new, heavier inflow, and the water level stabilizes.
Okay, I follow the physics there. So there's no sensor measuring the lake's depth and actively deciding to open a dam. The new stable water level is just an emergent property of the system.
Yes.
The balance simply settles where the input and output naturally equilibrate based on the laws of physics.
Exactly. Or rather, that's the foundational mechanism. We can apply this directly to something like body weight. Let's say you drastically increase your daily food intake. You're taking in more calories, so your body weight increases.
Sure.
But here's the biological equivalent of that hydrostatic pressure. As your body mass increases, the baseline energy cost of carrying, heating, and sustaining that extra mass also goes up.
Oh, because there's just more of you to move around.
Right. Your basal metabolic rate increases simply because there's more tissue to keep alive. Therefore, eventually, your newly increased daily energy expenditure naturally matches your higher caloric intake. Your body weight settles at a new higher point. There's no ideal weight sensor in your brain sounding an alarm. It's a settling point driven by the physics of energy costs.
Okay, I can see how that passive settling process works for body weight over a period of, months. But body temperature or blood pressure, those require split-second critical control. If my blood pressure just passively settled over a few weeks, I would pass out every time I stood up.
Right, yeah.
There has to be something more active going on, doesn't there?
You're bringing up the exact limitation of a purely passive settling point, which is why the modern consensus is actually the distributed settling point model.
Distributed settling point.
Yeah. It combines the active neural feedback loops you were thinking of with that emergent settling concept.
Yeah.
So let's look at body temperature again. You do not have one master error detector. Instead, you have a vast array of multiple independent temperature detectors scattered throughout your entire organism.
Really? Scattered everywhere?
Yes. You have thermal sensors in your skin, distinct sensors deep in your abdominal organs, and entirely separate sensors clustered in different regions of your brain and spinal cord.
So the detection is massively decentralized.
Highly decentralized. And here's the crucial part. Each of these different sensory populations is coupled through distinct parallel neural pathways to completely different effectors.
What do you mean by effectors?
Like the mechanisms that actually change the temperature. You have one pathway entirely dedicated to controlling the dilation of the blood vessels in your skin. You have another pathway wired just to your sweat glands,
Oh, wow.
another for the metabolism of brown fat, another for the mechanical action of shivering, and even parallel pathways driving behavioral changes, like the sudden urge to curl up into a ball or put on a sweater.
So when I step out into the cold, it isn't one master alarm going off. It's dozens of independent loops all firing simultaneously.
Yes, when your temperature fluctuates, these multiple separate sensory effector loops all engage independently. So that incredibly stable 37 degrees Celsius that we observe on a thermometer is not a programmed target.
Huh.
It's the emergent settling point. It's the result of the combined simultaneous activities of all these multiple feedback-informed loops pushing and pulling against each other until the system finds equilibrium.
That requires a total paradigm shift in how we view ourselves. I mean, we're not a machine built with a master blueprint of ideal numbers. We are a massive distributed negotiation of sensors and reactions that just happen to balance out at a state that keeps us alive.
It's arguably one of the greatest achievements of evolution. And this nuanced, distributed model applies across the board to temperature, blood pressure, blood osmolarity, and energy regulation.
But I mean, even a distributed negotiation needs a forum, right? These signals can't just float randomly in the ether. The data from the skin, the deep organs, and the spinal cord has to go somewhere to be integrated, prioritized, and coordinated.
Absolutely.
So we need to move physically into the brain to find the conductor of this massive physiological orchestra.
And that conductor is a region called the hypothalamus. It serves as the critical integrating center. It basically takes in the continuous status updates regarding your physiological parameters and translates that data into coordinated outputs directed at your behavioral, autonomic, and neuroendocrine motor systems.
Okay, let's map this out for the listener. If we were navigating a miniature submarine through the human brain, where exactly are we parking to find the hypothalamus?
We need to go deep into the base of the brain, situated almost perfectly in the center of your skull, immediately above the pituitary gland.
Why there?
Well, the location above the pituitary is not an accident. It's essential because the pituitary is the master gland of the endocrine system, and the hypothalamus needs direct access to regulate hormones.
Oh, okay.
It's a relatively small structure, but it's densely packed and anatomically bordered by major neural highways. It sits below the thalamus, which makes sense because the thalamus is the brain's primary sensory relay station, and the hypothalamus needs to tap into those sensory streams.
It sounds like a heavily guarded control room packed with different specialized units. But within this dense cluster, there's a specific region called the lateral hypothalamic area, or the LHA. It spans a large portion of the hypothalamus, but its job description seems radically different from the rest of the area. I mean, it doesn't seem to care about homeostasis at all.
Yeah, the LHA is a fascinating diversion from strict physiological balancing. It's linked much more closely to behavioral arousal and reward pathways.
Reward pathways, like dopamine.
Exactly. If you trace its connections, the LHA is heavily wired into the nucleus accumbens and the ventral tegmental area, which are the classic dopamine-heavy reward centers of the brain. Furthermore, the LHA contains specialized neurons that produce a neuropeptide called orexin,
Orexin.
also known as hypocretin.
Right.
Orexin is absolutely critical for stabilizing wakefulness. If you lose those orexin neurons, you develop narcolepsy.
Oh, wow. So you just fall asleep.
Right. So while other parts of the hypothalamus are quietly balancing your blood pressure, the LHA is driving the arousal and motivation required to interact with the outside world and seek out rewards.
Okay, so if the LHA is the motivation and arousal department, where are the actual homeostatic regulators? How do we know which specific clusters of cells are controlling temperature versus thirst? Because if the hypothalamus is so tiny and densely packed, it must be a nightmare to tease apart what does what.
Historically, it was incredibly difficult. Lesion studies where researchers would accidentally or intentionally damage a part of the brain and see what function was lost, they were just too blunt.
Yeah, like hitting a computer with a hammer to see what breaks.
Exactly. But recent advances using techniques like optogenetics and chemogenetics have revolutionized our understanding.
Let's pause there, because optogenetics is a term that sounds like science fiction. What does that actually mean in a laboratory?
It is brilliant. Researchers can use genetic engineering to insert light-sensitive proteins, similar to the proteins found in the retina of your eye, into very specific, targeted populations of neurons in a living animal.
Wait, like from an eye?
Yeah, literally light-sensitive proteins. And once those proteins are expressed in the cell membranes, scientists can shine a microscopic laser light onto that brain region. The light causes the proteins to change shape, which opens ion channels, causing those specific neurons to fire action potentials.
You can control an animal's brain cells with a flashlight.
Effectively, yes. You can turn specific subsets of neurons on or off with light, or in the case of chemogenetics, with designer drugs that only affect engineered receptors. By using these highly precise tools in active behaving animals, researchers have finally mapped out what we call modality-specific neurons.
Modality-specific, meaning what?
These are distinct anatomical clusters dedicated to governing very specific physiological parameters.
So they finally isolated the temperature cells from the thirst cells.
Yes. For example, the key regulatory neurons that drive the defense of body temperature are highly localized in a cluster called the median preoptic nucleus, or MNPO.
Yeah.
If we look at water balance, the regulation of your blood osmolarity, and the intense drive of thirst, those functions are controlled by neurons not just in the median preoptic nucleus, but also in specialized regions that actually lack a normal blood-brain barrier.
Wait, why would they lack a blood-brain barrier? Isn't that dangerous?
It's a calculated risk. They lack the barrier, allowing them to directly taste the saltiness of the blood. These include the vascular organ of the lamina terminalis and the subfornical organ.
Oh, wow. They're literally sampling the blood.
Exactly. And if we look at energy balance, the calculation of fat stores and the hunger drive, those are governed by distinct populations of neurons in the arcuate nucleus.
So even though homeostasis is a distributed settling point system playing out across the entire body, the sensory information from the body eventually funnels into these highly specific, dedicated neuronal clusters in the hypothalamus to coordinate the massive reaction.
Right. They serve as the critical bridge. They link the interoceptive sensory feedback, the continuous stream of signals reporting the status inside your body, with the motor outputs that drive adaptive behaviors like seeking water and physiological responses like constricting blood vessels.
But see, this brings us to a concept that completely changes how we view this system. We've been talking entirely about feedback. The body gets cold, the sensors detect the cold, the brain reacts to the cold. It implies the brain is always playing catch-up.
Right, waiting for a problem to happen.
Yeah. But the hypothalamus doesn't just react to what has already happened. It uses a mechanism called top-down feed-forward input.
Yes. This is perhaps one of the most elegant and predictive features of human neurobiology. The brain is not just a passive receiver. It is an active prediction engine. It anticipates future homeostatic challenges based on environmental cues, and it alters your physiology before the crisis ever actually occurs in your blood chemistry.
I need a concrete example of this. Like, how does the brain predict a physiological crisis before it happens?
Let's look at the arcuate nucleus, which we just established controls energy balance and hunger. Within the arcuate nucleus, you have a specific population of hunger-promoting neurons. When an animal is food-deprived, their fat stores drop, and their blood glucose drops.
Makes sense.
The feedback mechanisms detect this, and these hunger-promoting neurons begin to fire at a frantic high rate. This high firing rate translates psychologically into an aversive, uncomfortable deficiency state.
It feels bad to be starving.
Literally, it feels bad, and that negative feeling drives the animal to frantically seek food.
Right, but that's standard feedback. The fuel is low, the alarm rings. Where does the feed-forward anticipation come in?
It happens the moment the animal simply detects cues that predict the availability of food. If that starving animal smells a meal, or sees it, or even hears the sound of a food dispenser long before a single calorie has been ingested or digested, there is a rapid, dramatic drop in the firing rate of those hunger-promoting neurons.
The hunger neurons shut down just from smelling the food.
Almost instantly. The alarm bells are silenced long before there are any actual changes to the blood chemistry, the glucose levels, or the energy stores that the feedback systems are supposedly monitoring.
That is wild. The brain is effectively predicting the future to prevent a catastrophic overcorrection. It's like, okay, imagine if I'm freezing cold and I see a blazing campfire in the distance. I stop shivering, and I might even take off my heavy jacket while I am still walking toward the fire, rather than waiting for my internal core temperature to actually rise.
Right.
Because if I waited until my deep organs warmed up to take off my jacket, I would be standing next to a fire in a heavy coat and I would instantly overheat.
That analogy captures the essence perfectly. The brain knows the caloric intake is imminent. The process of eating, digesting, and absorbing nutrients into the bloodstream is incredibly slow.
Right, it takes hours.
Exactly. If the hypothalamus waited for the slow process of digestion to finally restore blood glucose levels before it turned off the hunger drive, the animal would gorge itself continuously for an hour. It would consume far more calories than it actually needed, causing a massive, dangerous homeostatic swing in the opposite direction.
Like massive blood sugar spikes and gastrointestinal distress.
Yes. By utilizing top-down feed-forward control, the brain prepares the body for the anticipated challenge and shuts off the drive preemptively.
And silencing those alarms has a huge psychological impact too, doesn't it?
Oh, absolutely. It ties directly into learning and motivation.
Yeah.
Because that high activity in deficiency-driven neurons is deeply aversive, rapidly countering that aversive state the moment a cue is detected, provides a powerful, rewarding teaching signal to the rest of the brain.
It feels good when the hunger fades just from seeing the food.
Yes. That relief reinforces the specific behaviors, the foraging, the hunting, the navigation that led the animal to find the food in the first place, ensuring it can repeat that successful behavior tomorrow.
Wow. Okay, so we've established the hypothalamus as this master coordinator. It integrates sensory feedback, it predicts the future with feed-forward input, and it makes the executive decisions. But the hypothalamus itself is just a tiny cluster of neurons buried in the skull.
Right, it doesn't have hands.
Right. It can't physically reach out and constrict a blood vessel in your leg. It can't force a sweat gland in your forehead to secrete water. How does it actually execute its orders across the vast geography of the human body?
To execute these vital homeostatic commands, the hypothalamus relies heavily on its dedicated motor arm, which is the autonomic motor system.
We hear the word autonomic thrown around constantly, usually as a synonym for automatic or involuntary. But anatomically, how does this differ from the somatic motor system, which is the system I use to consciously pick up a pen or go for a run?
The distinction lies in both the targets and the architecture. The somatic motor system has one job. It controls striated skeletal muscle. Those are the muscles attached to your bones that govern voluntary movement.
Biceps, triceps, quads.
Exactly. The autonomic motor system is entirely distinct. Autonomic motor neurons project out to regulate smooth muscle, which is the muscle lining the walls of your blood vessels, your respiratory airways, and your entire gastrointestinal tract.
Oh, I see.
It also controls the cardiac muscle of your heart, the gland cells serving both endocrine and exocrine functions, and critical metabolic targets like adipocytes, which are your fat cells.
So while the somatic system moves the skeleton through the environment, the autonomic system manages the entire hidden infrastructure keeping the organism alive.
Well said.
But I imagine it's not a dictatorship. Is every single autonomic function completely subservient to the hypothalamus?
No, and that is a vital nuance. The autonomic system is highly sophisticated and is also heavily regulated by local, lower-level circuits distributed throughout the brainstem and the spinal cord.
Oh, really?
Yeah. Because of this, different physiological functions vary drastically in their reliance on top-down hypothalamic commands.
Yeah.
For instance, thermogenesis, the production of heat by burning brown fat, is intensely dependent on direct hypothalamic intervention. However, a process like micturition, which is the technical term for urination, it's largely organized by local reflex circuits in the lower spinal cord and operates with a high degree of independence from the hypothalamus, though higher brain centers still provide the conscious permission to initiate the process.
Let's break down the physical architecture of this autonomic network. Following the classic nomenclature established by the physiologist J.N. Langley, the system is divided into three main branches. What are we looking at?
The three divisions are the sympathetic, the parasympathetic, and the enteric systems. Let's start with the sympathetic and parasympathetic divisions, as they share a general organizational principle. Unlike the somatic system, where a single long motor neuron stretches all the way from the spinal cord to the bicep, these autonomic branches use a two-neuron relay system.
A two-neuron relay. Break that down for me.
You start with a preganglionic neuron. Its cell body is located inside the central nervous system, either in the brainstem or the spinal cord. This preganglionic neuron sends its axon out to a peripheral ganglion, which is basically a junction box located outside the spinal cord.
Okay, so it stops halfway.
Right. Inside that junction box, it synapses onto a postganglionic neuron. This second neuron then travels the rest of the way to physically innervate the target organ.
Okay, let's trace the sympathetic division first using that two-neuron model. Where do the preganglionic neurons start and where do they go?
The preganglionic neurons for the sympathetic system are exclusively localized in the middle section of the spinal cord, specifically extending between the first thoracic segment and the third lumbar segment.
So the mid-back.
Right. Because of this specific origin, it is technically referred to as the thoracolumbar outflow. These preganglionic neurons are the foot soldiers, but they take their marching orders from descending supraspinal control, meaning signals coming from above the spine from higher brain areas.
Like where?
Important supraspinal structures driving the sympathetic output include the rostral ventrolateral medulla, the raphe pallidus, and the paraventricular nucleus of the hypothalamus.
But wait, why are they all clustered in the middle of the back? If they need to control things in the head and the feet, why not start everywhere?
It's an architectural design built for rapid systemic activation.
Mm.
By exiting the spinal cord in the middle, these preganglionic axons can immediately enter the sympathetic chain ganglia, a massive interconnected highway of junction boxes running parallel to the entire length of the spine.
Oh, like a parallel highway.
Exactly. Once the signal hits that highway, it can rapidly radiate upward to the head and downward to the legs. From these ganglia, the postganglionic neurons project out to innervate targets throughout the entire body.
What kind of targets are we talking about?
The reach is massive. We're talking about the smooth muscle lining nearly every blood vessel in your body, the sweat glands in your skin, the erector pili muscles that cause your hair to stand on end when you are cold or frightened.
Goosebumps.
Yeah, goosebumps. Your brown fat stores, and even the immune system, projecting directly into the spleen, the thymus, and your bone marrow.
Wow, that covers the hardwired neural connections. But the sympathetic system also has a chemical trick up its sleeve, right? It connects to the adrenal glands.
Yes, and this is a crucial exception to the two-neuron rule we just talked about. A very special subset of these sympathetic preganglionic neurons bypasses the peripheral ganglia entirely. Their axons travel straight to the adrenal gland, which sits on top of your kidneys, and synapse directly onto specialized cells in the adrenal medulla called chromaffin cells.
And when those chromaffin cells receive the signal, what happens?
They dump massive amounts of epinephrine and norepinephrine, adrenaline and noradrenaline directly into the general bloodstream.
Ah, so it becomes a hormone.
Exactly. Instead of sending a private electrical message down a wire to a specific organ, this turns the sympathetic signal into a systemic hormone. The blood carries the adrenaline to every corner of the body simultaneously, amplifying the sympathetic response and allowing it to exert powerful metabolic effects on distant targets that might not even have direct neural innervation.
So that explains the full-body rush of an adrenaline spike. So that is the sympathetic division, broad, systemic, originating in the middle of the spinal cord. What about its counterpart, the parasympathetic division?
The parasympathetic system has a completely different anatomical origin. It arises from the two extremes of the central nervous system: a cranial zone located up in the brainstem, and a sacral zone located at the very bottom of the spinal cord. Therefore, it is often called the craniosacral outflow.
So it skips the middle of the back entirely. Where do the signals go from the brainstem?
The cranial preganglionic neurons travel outwards, packaged inside 4 specific cranial nerves: the oculomotor nerve controlling the eyes, the facial nerve, the glossopharyngeal nerve, and most famously, the vagus nerve.
Ah, the vagus nerve. I mean, it's become somewhat of a celebrity in pop science lately. People talk about vagal tone constantly. Just how far does this one nerve reach?
Well, its name literally means the wanderer in Latin, and it absolutely earns the title. The vagus nerve, which is cranial nerve number 10, provides massive parasympathetic outflow descending from the brainstem all the way down into the thorax and abdomen.
It just wanders down through everything.
Yeah, it directly innervates the heart, the lungs, the liver, the gallbladder, the pancreas, the stomach, and the upper portions of the small intestine. It is the primary conduit for the brain to slow the heart rate and promote digestion. Once you move past the upper intestines, the sacral portion of the parasympathetic system takes over, projecting from the base of the spine to supply the large intestine, the rectum, the bladder, and the reproductive organs.
So the sympathetic and parasympathetic divisions form this vast dual wiring system reaching almost every organ. But then we arrive at the third division, the enteric system. And the numbers here are just staggering. It's described as having as many as 100 million neurons completely dedicated to the gut.
The numbers are correct. The enteric ganglia form a massive, continuous, web-like network embedded directly within the walls of the gastrointestinal tract. It stretches seamlessly from your esophagus all the way down to your rectum, and it includes regulatory control over the pancreas and the gallbladder. It is, by a massive margin, the largest and most complex division of the autonomic nervous system.
With 100 million neurons, it's often referred to as the second brain. But is that an accurate label? Like, if it has that much processing power, does it operate independently of the hypothalamus and the brainstem? If you completely severed the vagus nerve, would the gut just stop working?
From a purely functional standpoint, Calling it a second brain is a useful analogy to illustrate its complexity, though, we must be careful not to imply it possesses consciousness or intent.
Right, it's not writing poetry.
Right. But what truly sets the enteric system apart from the sympathetic and parasympathetic divisions is its intrinsic circuitry. The ganglia in the sympathetic chain are essentially just relay stations. A signal comes in from the spine and a signal goes out to the organ. But the enteric plexus is entirely different. It contains its own internal sensory neurons, its own inner neurons for processing, and its own motor neurons, all packed together in the gut wall.
Wait, it has sensory neurons that don't report back to the brain? They just report to other neurons within the gut itself?
Exactly. It forms complete, closed-loop reflex circuits, entirely localized within the tissue of the gastrointestinal tract. Because of this intrinsic circuitry, the basic fundamental functions of the gut-like peristalsis, which is the coordinated wave-like muscular contractions that push food through the intestines, they can actually be maintained even if you were to completely sever the splanchnic sympathetic nerves and the vagal parasympathetic nerves connecting the gut to the brain. The enteric system can govern the daily operations of digestion locally.
That is incredible. The gut can literally run its own factory without management, but the brain can't just ignore it, right? It still needs to know if the factory's out of raw materials or if there's a blockage.
Absolutely. The autonomy of the enteric system is not absolute isolation. While it manages the minute-to-minute operations locally, it is in continuous high-volume communication with the central nervous system. Through those same splanchnic nerves and the massive sensory afferent fibers of the vagus nerve which carry signals from the body up to the brain, the gastrointestinal tract is constantly bombarding the spinal cord and the brainstem with sensory updates regarding its distension, its chemical environment, and its progress.
So it's still sending up status reports.
Exactly. The central brain is always being updated and can override the local enteric circuits when necessary, such as shutting down digestion entirely during a physical threat.
We've covered the wiring diagrams, but we really need to talk about the actual language these nerves use to communicate. When a sympathetic nerve reaches the heart, how does the electrical signal cross the physical gap to actually make the cardiac muscle beat faster? The pharmacology here seems crucial.
Oh, it is the absolute foundation of modern neuropharmacology. To cross the synaptic gap, the neurons must release chemical messengers called neurotransmitters. Let's look at the two-neuron relay system again. At the first junction, in both the sympathetic and the parasympathetic systems, the preganglionic neuron, arriving from the central nervous system, releases the neurotransmitter acetylcholine, or ACE.
Acetylcholine?
Yes. This acetyl crosses the microscopic gap and acts on specific receptors on the postganglionic neuron called nicotinic receptors.
So the first handshake is always acetylcholine meeting a nicotinic receptor.
Yes. And that binding produces very fast excitatory postsynaptic potentials, ensuring the electrical action potential continues down the second neuron. But the critical divergence happens at the final destination, when the postganglionic neuron actually innervates the target tissue, like the heart or the lungs.
How do they differ at the target?
Parasympathetic postganglionic neurons typically continue to use acetylcholine as their weapon of choice. However, at the target organ, the HAC does not bind to nicotinic receptors. It binds to muscarinic receptors.
What's current?
Right. These receptors trigger completely different intracellular cascades, often slowing things down, like decreasing the heart rate. Sympathetic postganglionic neurons, on the other hand, switch their chemistry entirely. They primarily release norepinephrine, which acts on alpha and beta adrenergic receptors embedded in the target tissue.
So norepinephrine hits the beta receptor.
Exactly. And when norepinephrine hits a beta receptor in the heart, it accelerates the heart rate and increases the force of contraction.
But biology is never that simple, is it? Do they really just release one single chemical at a time?
You are right to be skeptical. The classic one neuron, one neurotransmitter model is a significant oversimplification. Postganglionic neurons frequently utilize a mechanism called co-transmission. They release their primary fast-acting neurotransmitter alongside a larger, slower-acting neuropeptide.
Okay, what does that look like in practice?
Well, for example, parasympathetic nerve terminals often release acetylcholine simultaneously with vasoactive intestinal peptide, or VIP. The ACA might cause a rapid, brief contraction of a muscle, while the VIP initiates a slower, sustained increase in local blood flow to support that muscle's metabolic needs.
And this detailed receptor pharmacology, knowing exactly where the muscarinic, nicotinic, and adrenergic receptors are located, is essentially the operating manual for vast swaths of modern medicine.
It really dictates the design of countless therapeutics. If a patient comes in with heart failure or dangerous hypertension, a cardiologist will often prescribe a beta blocker.
Ah, beta blockers.
Right. That drug circulates in the blood and specifically physically blocks the beta-adrenergic receptors on the heart muscle, preventing the sympathetic nervous system from overstimulating the heart. If a patient is having a severe asthma attack, their airways are constricting. They use an inhaler containing an albuterol compound, which is a beta-adrenergic agonist.
So it mimics the sympathetic signal.
Exactly. It mimics the action of the sympathetic nervous system on the lungs, commanding the smooth muscle in the airways to rapidly dilate so the patient can breathe. Understanding these autonomic synapses allows us to medically intervene in allergies, emphysema, overactive bladder, and cardiovascular disease.
This brings us to how these two vast systems, the sympathetic and parasympathetic, actually interact in our daily lives. Walter Cannon, who coined the term homeostasis, also gave the world the concept of fight or flight. Because of that, pop culture has adopted this binary view of the autonomic system.
Very binary, yeah.
We think of the sympathetic division as the ultimate gas pedal, like a singular switch you flip for pure panic and survival, and the parasympathetic division as the brake pedal, reserved only for deep sleep and digesting a heavy meal. They're framed as mortal enemies locked in a zero-sum war.
It is a pervasive misconception. Cannon's original insights into the adrenal response were brilliant, but they inadvertently created a paradigm that vastly overemphasized the extremes of autonomic behavior.
The extremes.
Yeah. He correctly identified that the sympathetic system promotes arousal, defense, and escape. And he observed that its actions can be highly diffuse, creating that whole-body adrenaline rush. He also noted that in those extreme scenarios, the sympathetic and parasympathetic systems act antagonistically. But what we now know is that during 99% of your everyday life, outside of sheer terror, these systems are not engaged in a tug-of-war. They are highly integrated, highly specific, and constantly cooperating.
So it's not a singular switch that floods the whole body every time I get slightly stressed.
Not at all. The sympathetic division is much more precise than Cannon envisioned. It contains highly specific, differentiated subsets of neurons that can control very targeted parameters independently without triggering a full fight-or-flight cascade. You can activate the sympathetic pathways to your brown fat to generate heat without simultaneously flooding your heart with adrenaline to run away.
How do they cooperate then? If they generally do opposite things, how do they work together?
Think of somatic motor control, like walking. To walk across a room, you do not simply tense every single muscle in your leg simultaneously. If you did, your leg would just lock up rigidly. To achieve fluid movement, you have to alternately flex and extend opposing groups of muscles in a highly coordinated hierarchical rhythm.
Right.
The autonomic systems do the exact same thing with our internal organs. Let's look at a concrete, everyday example of this coordination: the micturition reflex.
Bladder control. Let's break down the mechanics of that. How do the sympathetic and parasympathetic systems alternate to manage the bladder?
Micturition is a masterclass in hierarchical reflex organization. It operates in two distinct phases: a storage phase and an elimination phase. When the bladder is slowly filling with urine over several hours, you need it to act as a relaxed, compliant storage vessel, and you absolutely need the exit to remain securely sealed.
Very important.
Very. So during this filling phase, sympathetic tone is entirely dominant. Sympathetic efferent signals actively relax the detrusor muscle, which is the main smooth muscle layer of the bladder wall, allowing it to stretch and expand without increasing internal pressure. Simultaneously, sympathetic signals actively constrict the internal urethral sphincter, keeping the exit locked.
So the sympathetic system is proactively managing the storage, expanding the walls, and locking the door. Where does the parasympathetic system factor in?
Well, during the filling phase, the sympathetic system is also actively sending inhibitory signals to the parasympathetic ganglia, effectively putting a physiological clamp on them to ensure they do not accidentally trigger an emptying reflex prematurely.
It's holding the parasympathetic system back. What happens when the tank is full?
As the bladder fills, sensory stretch receptors embedded in the bladder wall begin to fire at an increasing rate, sending afferent signals up the spinal cord. When the tension reaches a threshold and higher supraspinal centers in your brain give the conscious green light that is socially appropriate, the autonomic balance shifts completely.
The balance of power flips.
Yes, the sympathetic inhibition is rapidly lifted. Parasympathetic tone immediately becomes dominant. The parasympathetic pathways now drive a powerful contraction of the detrusor muscle to expel the urine while simultaneously signaling the urethral sphincter to relax and open. It is a perfectly coordinated timed cycle of alternating dominance, much like flexing and extending a joint to take a step.
That reframes the relationship completely. They are dance partners, not enemies. Let's examine another reflex, one that has to happen much, much faster than bladder control. We briefly touched on blood pressure earlier. I think everyone has experienced this. You've been sitting on the couch watching a movie for 2 hours, the doorbell rings, and you stand up instantly. For a split second, your vision tunnels, your head feels light, and the room spins. That is orthostatic hypotension, correct? Gravity has suddenly pulled the blood out of your brain.
Yes. When you transition rapidly from supine or sitting to standing, gravity causes a significant volume of blood to pool in your lower extremities. This results in a sudden, dangerous drop in arterial blood pressure reaching your brain. If your autonomic nervous system did not detect and correct this instantly, cerebral perfusion would drop so low that you would lose consciousness and collapse.
So when I feel that momentary dizziness, what exactly am I feeling? Am I actively perceiving the millisecond lag in my autonomic nervous system trying to catch up?
You are experiencing the precise mechanical delay of the baroreceptor reflex arc. Here is the step-by-step breakdown of what is happening in the dark while you are grabbing the wall to steady yourself. You possess highly specialized stretch receptors, known as baroreceptors, embedded in the walls of the carotid sinus, which is in your neck, supplying blood to your brain, and in the aortic arch, right above your heart.
The actual physical stretching of the blood vessel is the sensor.
Exactly. When your blood pressure is normal, the blood pushes against the vessel walls, stretching them, and these baroreceptors fire a continuous baseline stream of afferent sensory signals to the brain. But when you stand up and the blood pressure suddenly drops, the physical stretch on the vessel walls decreases. Because there is less stretch, the baroreceptors drastically slow down their firing rate.
So a drop in blood pressure results in a sudden drop in sensory noise. Silence is the alarm.
That is a great way to conceptualize it. This sudden reduction in afferent signaling travels up to the brainstem and is instantly detected by a specific processing center in the medulla. The medulla registers the drop in pressure and immediately orchestrates a two-pronged, highly coordinated autonomic counterattack. First, it reflexively suppresses parasympathetic outflow to the heart. Remember, baseline parasympathetic activity via the vagus nerve constantly acts as a brake, slowing your resting heart rate down. By lifting that brake, the heart rate can instantly accelerate.
The vagal brake comes off.
Simultaneously, the medulla massively stimulates sympathetic efferent outflow. This sympathetic surge executes three critical actions at once. It further drives up the heart rate, it fundamentally increases the physical contractility, the raw squeezing power of each individual heartbeat, and most importantly, it causes widespread arterial vasoconstriction, aggressively narrowing the blood vessels throughout your body, particularly in your legs and abdomen.
Which increases the hydraulic resistance in the pipes.
Yes. The combined effect of the heart pumping more blood, pumping it with greater force, and forcing that blood into a narrower system of pipes rapidly spikes your systemic blood pressure. This hydraulic pressure forces the blood back up against gravity, restoring perfusion to your brain, and your vision clears. All of this happens in a matter of a few heartbeats.
And there is an important detail about the timing of these two systems working together. They operate at slightly different speeds, don't they?
This is a vital characteristic of autonomic cooperation. The parasympathetic component of the baroreceptor reflex, taking the vagal brake off the pacemaker cells in the heart, operates on a millisecond timescale. It has an incredibly rapid onset, but its effects are relatively brief. The sympathetic component, constricting the distant vascular beds and ramping up the contractility of the cardiac muscle tissue, takes slightly longer to reach peak effect due to the slower second messenger signaling cascades at the adrenergic receptors. However, the sympathetic system is responsible for the sustained, long-term regulation required to keep your blood pressure elevated for as long as you remain standing. They cooperate seamlessly in both physiological function and temporal execution.
Okay, this creates the perfect bridge to our final major topic today. We have explored the hypothalamus predicting challenges and sending commands downward. We have explored the massive sympathetic, parasympathetic, and enteric networks physically executing those commands on the organs. But for reflexes like the baroreceptor reflex to function, for the brain to know that blood pressure has dropped or that the bladder is full, the brain must receive a continuous, massive data stream from the carotid sinus, the stomach, and the deep tissues. How does the body talk back to the brain? Where do all these visceral sensory pathways lead? What is the first point of contact inside the central nervous system?
The primary gateway, the central hub for all incoming visceral sensory information, is a highly specialized structure located in the medulla oblongata called nucleus of the solitary tract, commonly referred to as the NTS. Almost all primary afferent sensory data from your internal organs, whether it is the mechanical distension of your gut, the chemical composition of your blood, or the pressure in your arteries, arrives first at the NTS.
So all the data funnels into the NTS in the brainstem. Once it arrives, does the NTS send it straight up to the conscious cortex so I can think about it? Like, oh, my blood pressure is low, I should fix that.
In most cases, absolutely not. If you are consciously aware of every minor fluctuation in your gut chemistry or your vascular resistance, the cognitive overload would paralyze you. The NTS processes and routes this incoming sensory information in two distinctly different directions. The first pathway is entirely local. The NTS projects directly to adjacent neural networks within the brainstem and the upper spinal cord. The purpose of this local pathway is to coordinate rapid subconscious autonomic reflexes without bothering the higher brain.
Like a local circuit breaker in your house that trips without you having to call the power company.
Precisely. This local circuitry is how visceral sensory signals arriving at the NTS directly regulate the vagal motor control of your resting heart rate or govern the rhythmic peristaltic contractions of your gastrointestinal tract. For example, some specialized neurons within the NTS project directly to the ventrolateral medullary reticular formation, the exact region we discussed earlier, to subconsciously manipulate your blood pressure by constricting or dilating peripheral vascular beds.
Okay, so the local route handles the automated subconscious reflexes. What is the second direction the NTS sends data?
The second direction involves ascending projections that travel upward into the forebrain. This is how vital visceral information reaches the higher, more complex structures of the brain, allowing the hypothalamus and the cerebral cortex to orchestrate highly complex neuroendocrine, emotional, and behavioral responses to internal states.
How does the signal travel up to the forebrain? Is it just one long, direct fiber?
There are some direct pathways, notably direct projections from the NTS straight into the hypothalamus. But the primary high-volume route is an indirect pathway. The NTS sends its efferent fibers upward to a relay station called the lateral parabrachial nucleus. From the parabrachial nucleus, the sensory information radiates broadly outward to highly sophisticated forebrain structures. It projects into the amygdala, the bed nucleus of the cerebral cortex, the hypothalamus, and critically to specific areas of the cerebral cortex, most notably the insular cortex and the medial prefrontal cortex.
This is where things get philosophically profound for me. We traditionally view the prefrontal cortex as the seat of our highest humanity. It is where we process complex logic, abstract reasoning, and long-term planning. But according to the physiological mapping, the medial prefrontal cortex, specifically the anterior cingulate gyrus and the prelimbic areas, is actually a visceral sensory motor region. If researchers electrically stimulate these specific high-level cortical areas, they can physically cause a subject's stomach to contract or their blood pressure to spike.
It forces a complete reassessment of how we view the hierarchy of the brain. These advanced cortical regions are not floating above our biology, they are deeply embedded in it. They constantly receive visceral sensory input, they process the emotional and cognitive context of that input, and then they send powerful, descending, top-down motor commands all the way back down the neuroaxis to the central autonomic network in the brainstem.
This means our highest cognitive processors are physically, anatomically hardwired directly into our most primitive, lower-level gut reflexes. If I am understanding this correctly, does this anatomical pathway provide the literal, physical explanation for psychosomatic symptoms? Does this neuroanatomy explain why experiencing severe, chronic psychological stress at work, like worrying about a complex social conflict or a financial problem which is processed in the prefrontal cortex, can physically destroy our digestion or cause chronic hypertension?
It absolutely does. It completely eliminates the artificial separation between the mind and the body. It provides the exact, mapped anatomical highway demonstrating how cognitive, emotional, and psychological states exert direct physical control over your internal organs. When you experience chronic cognitive stress dwelling on abstract anxieties in your prefrontal cortex, that region does not just spin its wheels in isolation, it is directly wired downward into the central autonomic network. The prefrontal cortex sends cascading, descending signals that can artificially alter your resting vagal tone, chronically elevate sympathetic outflow, and directly disrupt the delicate balance of the 100 million neurons operating in your enteric nervous system. The stress in your mind travels down a paved physiological highway to alter the acid secretion and motility of your stomach.
The connection isn't metaphorical or spiritual. It is literal neural wiring.
Yes. And we see similar high-level integration in a midbrain structure called the periaqueductal gray, or the PEG. The pega receives massive inputs from the cortex, the amygdala, and the hypothalamus. Its primary role is to coordinate and initiate highly complex, integrated whole-body survival responses that perfectly match autonomic output to behavioral action.
How does the PEG integrate behavior and autonomic control?
Consider a severe defensive scenario, a true fight-or-flight emergency where you must physically defend yourself. The PEI does not just generically activate the sympathetic nervous system and hope for the best. It orchestrates a highly specific hemodynamic symphony. It initiates the behavioral motor programs for defense or fleeing, and simultaneously, it specifically commands the autonomic system to actively redirect blood flow away from non-essential systems like your digestive tract and your skin, and forcibly routes that high-pressure blood directly into the skeletal muscles of your hind limbs. It proactively optimizes your internal hydraulics to maximize your physical ability to fight or run. It ensures your physiology perfectly matches your behavior.
We have traced an incredibly massive continuous loop today. We began by grappling with the pure physics and philosophy of homeostasis, realizing that our biology is not trying to hit a static programmed number, but is constantly fighting to maintain a dynamic emergent settling point amidst the chaos of the external environment. We journeyed deep into the skull to find the master control room in the hypothalamus, a structure that not only monitors our present state, but actively predicts future crises using feed-forward anticipation. We followed the descending commands down the efferent autonomic pathways, mapping the sympathetic chains, the wandering vagus nerve, and the massive autonomous network of the enteric second brain, and we saw how these systems constantly cooperate in a delicate dance rather than fighting each other. And finally, we followed the sensory signals back up through the nucleus of the solitary tract, ascending all the way to the medial prefrontal cortex, proving anatomically that our highest, most abstract thoughts are inextricably physically linked to the primitive rhythms of our gut and our heart.
It is a staggering feat of biological engineering. When you understand the sheer density of the pathways, the speed of the synaptic relays, and the complexity of the distributed feedback loops, you realize that there is an invisible, silent orchestra playing at maximum intensity inside of you every single second, just to keep you upright, breathing, and free from the constraints of your environment.
It is awe-inspiring, but there is a crucial limitation to everything we have discussed today. The autonomic nervous system, the reflex loops, the action potentials firing down the vagus nerve, all of this operates primarily on rapid electrical signals. It is built for split-second adjustments. Milliseconds matter when you stand up and your blood pressure drops. But long-term survival requires more than just rapid electrical reflexes. What happens when the brain needs to mandate massive, systemic, long-lasting changes? What happens when it forgoes the electrical wires and instead dumps slower, incredibly powerful chemical messengers directly into the bloodstream?
That shifts the paradigm entirely. That brings us into the realm of the neuroendocrine system and the profound influence of circulating hormones.
And that is exactly where we are going next. In part 2 of our deep dive into the neurobiology of survival, we are going to look at how this neuroendocrine system literally controls our deepest deficiency drives. We are going to explore how the brain translates cold chemical imbalances in the blood into overwhelming, unavoidable behavioral urges. We will see how the chemistry of your body forces you to experience the intense, conscious motivation of thirst and hunger. We will see how that invisible autopilot doesn't just quietly steer the plane in the background, it actively dictates the behavior and the desires of the passengers. Until then, take a moment to pause and appreciate the immense, silent, distributed machinery working tirelessly to keep you alive right now.